Okay, last quick video on transcription and translation. So we've gone through all the different parts here about gene to protein. We covered transcription, we covered translation, and these are just kind of reviews, right? So we looked at how polymerase reads along that thing in 5' to 3', and we learned about introns and exons, and we learned about spliceosomes and SNRPs. We also learned about the promoters and the transcription factors, which really help all of this work along. This is the kind of nitty-gritty detail. Genes don't just get transcribed. A lot of things have to fall into place in order for them to actually get transcribed. And then we went into translation, like how that ribosome actually works. And then we talked about these initiation and elongation and termination ideas so that the whole thing finishes out. And then we also just covered the idea of codon charts, and hopefully I didn't confuse it too badly because I had the wrong codon here for my stop codon, and we got our codons figured out. So reviewing the whole thing here, we have transcription that's going to take place in a nucleus, that you're going to make a strand of mRNA, and that's going to process from, or it's going to be made from 5' to 3', and then it's going to be processed. There's going to be exons and introns, and the introns get cut out. And then a poly A tail is put on there, and a 5' G cap is put on there, and then that gives it its ticket to leave the nucleus. It leaves the nucleus, and it goes and it finds a ribosome, which is made of 2 subunits. And the ribosome has an A, P, and an E site. And as those sites get read, so that as the tRNA arrives, so this is tRNA, it matches up its anticodon with a codon, and it brings an amino acid to the party. And when 2 amino acids are side by side, they prefer to be bonded together instead of to their tRNAs. And so the whole thing shifts over and it will eject. That's what the E site is for. It'll eject out the old tRNA. Now, what hasn't been covered yet, and just another little detail here, is that tRNA that's now naked, it's going to go floating out in the or in the cytoplasm, out in the cytoplasm. And there are amino acids always available because things eat proteins and they get amino acids from those proteins. And those amino acids need to be partnered up or paired up with their tRNAs. So there's an enzyme out here called aminoacyl tRNA synthetase. Quite a mouthful, but think about it. Amino acids and tRNA are synthesized together. And it ends in ACE because it's an enzyme. And so what it does is just holds onto this tRNA and it holds onto this amino acid, and click, it puts those two together. And then it's ready to go back to the party and bring an amino acid to the sequence if it needs it. Okay. Once you hit your termination phase, then the whole thing stops and the polypeptide is made. And remember that chain of amino acids is a protein now, and it folds, primary, secondary, tertiary, quaternary folding, and it folds up, and its form, its shape, determines its function. Okay? So in addition to the information in the diagram over here, can you explain and label a promoter region, a terminator? Where would that be in this diagram? What are they? How about SNRPs and spliceosomes? They're not in this diagram. Where would they be? What do they do? And then finally, the question is, if DNA, not mRNA, but if DNA was as follows, so if we had a DNA part that we're reading a code from, what would the amino acid sequence be for this thing? Okay, I'm going to let you pause this for a second, try to find out those answers, and I'll pause for about 5 seconds, and then I'll go ahead and dig in on the answers. Okay, so ultimately, hopefully you've paused it yourself and giving yourself some time to think about these. The promoter and the terminator regions would be located somewhere on the DNA strand. The terminator region would be somewhere downstream from where the polymerase is working, and the promoter region would be somewhere upstream. We're also failing in this diagram, we're failing miserably to show things like transcription factors and an enhancer region and a bending protein and activators. I mean, this diagram can't show everything, but those things are usually involved. You might wonder why all those things are necessary. Why are there so many stupid steps? The reality is that allows cells to regulate when they actually make the protein. You don't need to make a protein all the time. Let's say it's a protein for digestion. You don't always have to have it available. You wouldn't want it always available. You'd only want it when you get, food in your stomach or certain hormones that are telling you that like you've eaten some food. And so those hormones and those chemicals interact with your DNA and actually activate transcription. Okay. Where would SNRPs and spliceosomes be? They would be in this picture here. They would be in these little loops, right? The SNRPs would be those little proteins that are pulling this mRNA piece into a loop, and they would form a spliceosome, the overall structure that actually like clips and cuts that stuff and then splices it back together. And then finally here, the other question that I came up with is, if DNA, not mRNA, was as follows, what would the amino acid sequence be? What we have to do here first is we have to get this transcribed into mRNA. So I like to do this by just splitting this into codons to begin with, because it just makes things easier for me. So TAC, the codon for that, the complement of that, the mRNA that would come out of that would pair up here, would be AUG. And then the next codon would be GAG. If you can see that would be our codon there. And then the next codon would be, if I'm looking up here at this AAA, the codon that would be created there would be UUU, because you got to remember uracil instead of thymine. And then the final codon here would be UGA. And I'm going to take this thing, and I am actually going to cut it, and I'm going to bring it back to a different page here. So I can use my codon wheel here, and I'm going to insert it. I'll just pop it in over here, just so that we have that to look at. All right. So AUG, what is that going to give us? Well, if we look at either one, you can use the wheel or you can use the box. Depending on the test, they'll expect you to kind of know how to use either or. But AUG would be, like if I'm using the wheel here, this would be AUG, that would be methionine. So I would just put MET for this one. So I would write MET underneath here. That would be our amino acid. And what's interesting is that every protein always starts with methionine. That's always the start codon. That's why in this diagram over here, MET is green, because it's the go, it's the start. It's always the start. So AUG is always our start here. And then we move over to GAG, and we could use the wheel. I'll go back over here just to get some practice with that. There's G, first letter, U is the second letter, and then G is the third. That's going to be valine. And notice that it doesn't matter what the third letter was. GU and anything was going to give you valine. Okay, and that'll become more important here as we move forward. But for right now, we just know that that's valine. And then UUU, I'll go back to the wheel just to mix it up. UUU would be phenylalanine. So we just put phenylalanine on there. And then UGA, if you can imagine, UGA is stop. And again, if we did it over here, it would be UGA. There it is, stop. And that is the terminal sequence, and that just says stop making that protein. So our protein here that we've made is 3 amino acids long, met, val, phenyl, and then stop. And so that would be our short protein. In reality, proteins are very long. They would be at least, a couple hundred amino acids long, which means 3 nucleic acids for every amino acid. Think about that, 3 nucleic acids, like AUG, for every amino acid. So you're talking about very long sequences to read out, okay? But for practice, now we hopefully have that, and that is the review that we needed, okay? So if you guys have any questions, let me know. And other than that, thanks for watching.